dan aturan, dengan berjiwa Pancasila, bersikap adil jujur berkarya, dalam membangun putra putri bangsa. Politeknik Negeri Pontianak turut membangun generasi bangsa dengan mencipta tenaga profesional dalam mewujudkan cita-cita bangsa. Bersikap teguh dalam melangkah, tepat waktu, ukuran dan aturan, disiplin tinggi dalam berkarya, demi untuk menggapai prestasi, menciptakan generasi bangsa untuk membangun negeri dan bangsa, bertanggung jawab di dalam berkarya. Majulah Politeknik Negeri Pontianak, semoga abadi dan tetap jaya. Gerak melangkah mengejar cita, Politeknik Negeri berjiwa teguh dalam berkarya, mengejar cita-cita bangsa dan negara, siap mengabdi sepanjang masa. Politeknik Negeri Pontianak menjunjung tinggi tridharma, perguruan tinggi dengan semangat Pancasila, tepat waktu, ukuran dan aturan, dengan berjiwa Pancasila, bersikap adil jujur berkarya, dalam membangun putra putri bangsa. Gerak melangkah mengejar cita, Politeknik Negeri Pontianak berjiwa teguh dalam berkarya, mengejar cita-cita bangsa dan negara, siap mengabdi sepanjang masa. Politeknik Negeri Pontianak menjunjung tinggi tridharma,
Ada tapi tadi
tepat waktu, ukuran dan aturan, dengan berjiwa Pancasila, bersikap adil jujur berkarya, dalam membangun putra putri bangsa. mengejar cita, berjiwa teguh dalam berkarya, mengejar cita-cita, sepanjang masa, politeknik negeri Pontianak, negeri Dharma,
Ada respon. Nah, ada.
ukuran dan aturan, dengan berjiwa Pancasila, bersikap
Bisa bang, settingin mic doang bang, gak usah speaker. Speakernya nggak usah mic.
Maksudnya ini mic-nya suaranya kemana nih?
Ke zoom kan, yang speakernya nggak usah jadi. Soalnya kayak apa aja dia.
dengan semangat Pancasila,
Audionya pakai suara TV.
dengan berjiwa Pancasila,
Panjang nggak? Ini nggak kayak jadi. Ya dulu baru.
Kalau kita ngomong soal masih.
generasi bangsa, dengan mencipta tenaga profesional, dalam mewujudkan cita-cita bangsa, bersikap teguh dalam melangkah, tepat waktu, ukuran dan aturan, disiplin tinggi dalam berkarya, demi untuk menggapai prestasi, menciptakan generasi bangsa, untuk membangun negeri dan bangsa, bertanggung jawab di dalam berkarya, majulah Politeknik Negeri Pontianak, semoga abadi dan tetap jaya. Gerak melangkah mengejar cita, Politeknik Negeri Pontianak, berjiwa teguh dalam berkarya, mengejar cita-cita bangsa dan negara, siap mengabdi sepanjang masa, Politeknik Negeri Pontianak, menjunjung tinggi tridharma, perguruan tinggi, dengan semangat Pancasila, tepat waktu, ukuran dan aturan, dengan berjiwa Pancasila, bersikap adil, jujur, berkarya, dalam putra putri bangsa. Gerak melangkah mengejar cita, Politeknik Negeri Pontianak.
Ada, Pak.
Kalau di sini kan ini kan hanya nggak bisa ngomong gitu.
Pakai mic itu ya? Ada mic kecil emang?
Mic kecil.
Tapi kan ada pakai ini.
Iya ya.
Mic tapi sambutan harus kedengaran di sini.
Tapi nanti kalau saya pun nggak mau pakai mic ini gitu ya. Bukan mic SIM, mic ini bukan mic laptop.
Berarti kita coba juga yang dari sini-sini.
Tadi udah dicoba bisa kan tadi.
Coba. Tes. Ini masih pakai sama.
Namanya yang itu.
Tes.
Okay, good morning, everybody. Before we get started, now we are still waiting for our conference. Well, now we are checked out our mic for the Zoom and YouTube. Thank you so much for coming and the counting hall today, Thursday, 16 November 2023. Our program is the first international conference on agriculture, science, and technology, I think that's 2023. Sound ini dari Zoom jadi memang agak delay. Karena saya ngomong di sini, di sana kan nggak menyebut itu udah dari Zoom sama YouTube-nya lebih lambat dibanding di sini. Gak apa-apa ya Bapak Ibu ya. Jadi itu suara Zoom ya.
Yes,
Gerak melangkah mengejar cita, Politeknik Negeri Pontianak. Berjiwa teguh dalam berkarya, mengejar cita-cita bangsa dan negara, siap mengabdi sepanjang masa. Politeknik Negeri Pontianak menjunjung tinggi Tridharma, perguruan tinggi dengan semangat, tepat waktu, ukuran dan aturan, dengan berjiwa Pancasila, bersikap adil, jujur, berkarya dalam membangun putra putri bangsa. Derap melangkah mengejar cita, Politeknik Negeri Pontianak berjiwa teguh dalam berkarya, mengejar cita-cita bangsa dan negara, siap mengabdi sepanjang masa. Politeknik Negeri Pontianak menjunjung tinggi Tridharma, perguruan tinggi dengan semangat Pancasila, tepat waktu, ukuran dan aturan, dengan berjiwa Pancasila, bersikap adil, jujur, berkarya dalam membangun putra putri bangsa. Politeknik Negeri dalam berkarya demi untuk menggapai prestasi, menciptakan generasi bangsa untuk membangun negeri dan bangsa.
Welcome Ms. Roro Pangilinan, speaking and reading song, our speakers, and our moderator.
Yang muter Indonesia Raya kan nanti di sini kan? Saya berarti. Gitu ya?
Yang dimohon kepada anak-anak peserta juga kepada audiens semua, mungkin yang di belakang boleh mengisi bangku di depan terlebih dahulu supaya full. Yang di belakang mungkin boleh Bapak kasih isi dulu di depan supaya full. Ya, silakan yang di belakang maju aja ke depan. Di sini masih ada beberapa space.
Itu enggak ada
Jadi anak nanti untuk di depan
Indonesia Bapak enggak ada mic in sini.
Mic in, main on yang mana yang masuk? Coba, tunjukkan. Mic in, mic out di
Ada
Ah,
khusus untuk suara masuk, suara keluar gitu.
settingnya di mana ya, lupa saya.
full kan aja dulu.
Sini kah?
Jadi nanti pas dia
Di depan nih, Bang.
pick out, dia cuman pick out doang kayak pas Indonesia Raya kan takut suaranya mantul ke dalam mic Bapak untuk in kan. Jadi suara dalam zoomnya mantul ke dalam.
Mungkin Bang Budi juga paling depan Bang Panji,
Pokoknya tuh mic laptop.
Ibu,
Iya, itu
Bu Rizky.
Saya ngetes lagi ya.
mantul dia Pak.
Kita ini udah online, nggak bisa ngeliat. Tadi sih kan yang kita pakai dunia lain kan.
Di depan boleh.
Kalau kondisinya kemarin sudah ada mic ini,
Bu Rizky,
saya set
boleh
ya.
di depan.
Juga kemarin nggak minta langsung di
Bisa jadi profesi di silakan.
Jadi gimana? Jadi nanti di Indonesia Rayanya
Udah sih, Bapak ya sekali ya.
masalah mutar.
Kan Bapak di belakang kan tadi kan?
Ya?
Belakang kan Bapak nih. Belakang tetap sini.
Di sini terus. Udah kayaknya nggak pindah-pindah lagi di sini.
Oh iya, speakernya. Bisa tadi setting ini Pak.
Baru gini, kita muter Indonesia Raya, kita muter lagu Polnet ya. Tes.
Untuk tesnya boleh nanti.
Kita coba saya
Volume segini pusing nggak saya? Nggak volume mixer. Ada versi mixer.
saya ini nih, nggak familiar setting-setting mixer gitu.
Ini apa sih Pak? Itunya OS-nya?
Hah?
OS-nya apa?
Windows lah.
Windows 11 kah?
11 kayaknya, 11.
Oh, pantas lah.
Kenapa?
Nggak familiar.
Itulah.
Saya kan 10 masih pakai, belum pakai
Saya
11.
jangankan itu, saya sendiri aja tidak terlalu hafal.
Kita coba yang himne ya.
Maksudnya mau di share screen nih.
Oh, ya, share screen Bapak berarti ya.
Kan kalau kita mau itu bisa didengar gitu kan berarti di share screen.
Share screen.
Masalah ini.
Bentar. Mana background Bapak yang itu? Buka background
See,
Bapak. Background apa tuh yang tempat Bapak nyimpan background gimana?
Di icon face nih, icon face.
Iya.
Hmm.
Mana?
Nih.
Ini kah? Mana? Bisa kecilkan Pak. Oke. Sematika share screen saya ini kan Pak ya?
Yang dari lembaga udah datang nggak? Pak Wawan, Pak Irawan, belum ya? Jam 9 bro. Dan bisa sekarang saya share lagi ini udah berapa ratusan dah yang di dalam Zoom.
Tahu.
Terus kita juga nggak bisa bahas lagi suara masuk
see,
bisa ada choice lagi sih.
Enggak, tadi itu saya mau pakai yang ini kan foto tuh, cuma enggak.
yes. See,
Sebentar. Mixer di mana?
Mixer apa Pak?
Ntar coba. Yaudah sini. Mixer yang mana? Yang tadi kan? Yang ini kan?
Diapain Pak?
Nah ini
Mau diapain?
saya
Oh, suara
Zoom meeting ya.
ini udah 100% ya semuanya.
Meetingnya udah
suara yang kayak ini nih ke apa? Ke speaker di dalam. Cuman nanti harus besar kecilkan lagi pas lagu. Sebenarnya itu
dan pusing dan bikin sebenarnya kita.
Kayaknya sih udah pusing.
kalau dia harus kecilkan lagi, besarkan lagi agak
Masker
ribet
masuk
sih.
IT nggak ada.
Suara kecil.
Itu sih tinggal dikecilkan aja sebenarnya kalau tapi harus dibesarkan lagi masalahnya nanti pas lagu.
Kalau saya berarti kalau suara untuk masuk ke sini kan berarti harus pakai mic ya. Karena pakai mic aja kan untuk suara masuk ke sini ke Zoom kan.
Oh, mic ini bisa dimatikan nggak ya, coba ya?
Bentar, kenapa?
Satu masalah. Alah, nggak kalah aku ya.
Maksudnya apa nih yang
Nah itu mikirnya kami sendiri bisa nggak ada.
ini kan udah pakai ini kan? Ini kan udah nyambung ke mic.
Jadi mic level 9 itu yang laptopnya juga miknya harus ini.
Saya juga bingung sebentar. Sebentar. Coba ya, pesan Indonesia Raya gitu. Semoga
Dia masih kan?
Tetap bukan mic in kah?
Ini dua-duanya soalnya double. Kan mic-in nggak? Bisa mic sama bisa speaker juga. Coba dulu lah. Kalau nggak bisa juga baru kita coba mic-in. noise. Jadi solusinya
Before we get started, still check our sound. Thank you so much for coming at the accounting hall today, Thursday, 16th November 2023. It's our conference, first international conference on agriculture, science, and technology, Conference 2023. Sebelum kita memulai rangkaian acara kita, supaya tidak ngantuk ya, silakan boleh dibawa di tempat masing-masing.
Maksud, ya lah. Ini bagus. Udah. Tes. Oke. Tes. 1, 2, 3, Pak. Sini enggak keluar. Masuk. Aman. Iya, ini kan Zoom. Tes.
Di sininya enggak kedengaran, tapi sininya
Zoomnya kedengeran.
iya dari tadi masih mic lag.
Di sini. Tes, tes, 1, 2, 3. Tes 1, 2, 3. Masih belum sih Pak ya?
Dari awal tadi bisa.
Bisa. Mic-nya. Bisa di sini ya? Masalahnya mic Zoom-nya tinggal. Bisa. Bisa.
Gila ini.
Ini suara.
Belum. Boleh, boleh. Sebentar kan.
Baiklah, dimohon kepada para peserta untuk kembali ke tempat duduk masing-masing karena suara kita akan segera dimulai. Untuk proses acara kita pada hari ini, dimohon untuk mensinkronkan handphone atau kemudahan dan juga buat Bapak Ibu semuanya silakan mengambil snack dan coffee breaknya. Segera acara segera dimulai. Siap? Boleh. Assalamualaikum warahmatullahi wabarakatuh. Shalom. Good morning, ladies and gentlemen. Welcome to the first international conference on agriculture, science, and technology ICANN-FAST 2023. By Agriculture Technology Department, State Polytechnic of Pontiac. With them is the future of agriculture, exploring emerging technologies, scientific breakthrough, and sustainable practice. Give applause for our conference. Thank you so much. First, let me introduce myself. My name is Adela Iloga Saskia STRP, but you can call me Adela. I am an alumni from Agriculture Technology Department, State Polytechnic of Pontianak 2023. And now, I will be serving as your host as well for the next 6 hours today. It is an honor for me to inform you that around the past year, we have His Excellency Prof. Takuya Sugahara from Ehime University, Japan. His Excellency Dr. Samsul Haq from Charles Sturt University, Australia. His Excellency Pankrit Sohaipakar PhD from Fujian Agriculture and Forestry University, China. And Her Excellency Dr. Surisa Korovide from University Malaysia, Sarawak, Malaysia. His Excellency Dr. Haji Widodo PS FTMT, Director of Organization Polytechnic, and last, His Excellency Deputy Hendra Gunawan STMT, Head of Agriculture Technology Department. Well, the respectable all invited guests and also the honorable all participants of ICANN Fest 2023. So, before we get started, I will inform you the schedules of the conference today. This event will be divided into 4 mixed agendas, which are opening ceremony, plenary session that 4 keynote speakers will deliver their presentation, parallel session, and last, closing of ceremony. Dear participants, as it is hybrid the conference, I want to remind you who participate this conference online, please make sure you have a nice place with good internet connection. So you will get the best experience of this conference. Thank you so much. Dear all participants, without further ado, please stand for our national anthem, Indonesia Raya. Please stand.
Indonesia bersatu, hiduplah negeri bangsa semuanya semuanya padanya untuk Indonesia Raya. Indonesia.
Thank you so much. All participants, please have a seat. Now, let us involve the presence of our guest as Mr. Ade Sutawi leads us in the opening prayer. Prayer will be delivered Islamically and in Indonesian. Participants with other religions can adjust it. Mr. Ade Sutawi, time is yours.
يا ربنا لك الحمد كما ينبغي لجلال كريم وعظيم سلطانك. اللهم صل وسلم على سيدنا محمد وعلى اله وصحبه اجمعين. اللهم اسالك من تحمد لنا بك انتهينا يا من لم يبلغ السماوات والارض يا ذا الجلال والاكرام يا حي يا قيوم. ربنا ظلمنا انفسنا وان تغفر لنا وترحمنا لنكن من الخاسرين. اللهم اغفر لنا ولوالدينا وارحمهم كما ربيانا صغارا. واغفر لنا والنار والصراط الحياة بك يا أرحم الراحمين. اللهم اسقط الهدى والتقوى والغنى. اللهم اسقط من خير كل عاجل وآجل ما علمنا منه وما لم نعلم. ونعوذ بك من شر كل آجل وعاجل ما علمنا منه وما لم نعلم. اللهم اسقط من خير ما نسأل عبدك ونبيك. ونعوذ بك من شر ما علم عبدك ونبيك. اللهم اسقط الجنة وما قرب إليها من قول وعمل. ونعوذ بك من النار وما قرب إليها من قول وعمل. اللهم نسألك أن تجعلنا كل من كنت لنا خيرا من حرام. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار. وصلى على عبدك ورسولك النبي الأمي وعلى آله وصحبه وسلم. سبحان ربك رب العزة عما يصفون وسلام على المرسلين الحمد لله.
Thank you very much, Mr. Ali Subhani. Now let us move to the next agenda, which is speech by some speakers. The first opening speech from conference chair will be delivered by the secretary of this conference. Please welcome Ms. Jane Hillary, LSIMSI. The time is yours. Assalamualaikum warahmatullahi wabarakatuh. Shalom. Good morning, ladies and gentlemen. We are asked to deliver important messages from our chief of this conference, Mr. Jimmy Family MSI. We would like to welcome all of you to the first international conference on agriculture, science, and technology. It is with great pleasure and excitement that we gather virtually to join the fourth conference in these vital fields. This conference stands as a testament to the power of collaboration and shared history. It is not a great chance for us to collaborate with some exceptional keynote speakers from 4 different countries, from Rahwila Sudan, from AUB University, Japan, Dr. Samsul Haq, from Charlestown University, Australia, Hafiz Sohail Masar, PhD, from Fujian Agriculture and Forestry University, China, and Dr. Suisa Amkili, from University Malaysia Sarawak, Malaysia. We would like to extend our appreciation to the distinguished director of Oil, Gas, and Polytechnic, Dr. Haji Widodo, SFPN, for his unwavering support in making this event a reality. Furthermore, we would like to recognize the leadership of the head of the Department of Agricultural Technology, Mr. Desri Hendra Gunawan, SDMP. We also express our deepest gratitude to all oral presenters for some international campuses, auditors, and the division organizing committee. This session will showcase the most breakthrough and application in agriculture, science, and technology. The participants today may be coming from different backgrounds, academicians, practitioners, governments, and students. So we say thank you all for participating in this event. We encourage all of you to actively engage in discussions, forge new connections, and leverage this opportunity to broaden your horizons. Let us join hands making the International Conference on Agriculture, Science, and Technology a resounding success. Thank you and welcome to this incredible journey of knowledge and innovation. Wassalamualaikum warahmatullahi wabarakatuh. Shalom. Well, thank you so much, Miss Jean Hillary, as is I and Miss. Next event. The second line speech will be delivered by the Head of Agriculture Technology Department, Mr. Deputy Henry STMT. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Leslie Gunawan. I am the Executive Departemen SMI Indonesia. Pertama-tama, atas nama Ketua Jurusan Teknologi Pertanian, saya mengucapkan welcome to our international conference, khususnya kepada Bapak Direktur Politeknik Negeri Pontianak, Bapak Widodo PS, STMT. Pada hari ini diwakilkan oleh Bapak Ketua J1, Bapak Insinyur Haji Perawan. Yang saya hormati Ibu Linda Wajib Tua yang telah hadir. Yang saya hormati Profesor Nur Muhammad Wasna Politeknik Negeri Pontianak. Yang saya hormati Ibu Ika Yanti, Sekretaris Senat Politeknik Negeri Pontianak. Yang saya hormati Ibu Purnawati, Kepala Pusat Penelitian dan Pengabdian kepada Masyarakat. Yang saya hormati para tamu undangan. Yang saya hormati kepada Bapak Ibu Kepala Jurusan di lingkungan Politeknik Negeri Pontianak. Yang saya hormati para dosen, Bapak dan Ibu dosen di jurusan Teknologi Pertanian. Yang saya hormati para staf dan teknik di jurusan Teknologi Pertanian serta mahasiswa dan mahasiswi jurusan Teknologi Pertanian. Dan saya juga ingin mengucapkan thank you DKI Speaking pertama kepada Mr. Dr. Sabtu Bakri dari Australia. Berikutnya Mr. Faksis Sohib Masdar PhD dari Cina. Mr. Profesor Takuya Negara dari Jepang.
Isu tentang pertanian
Dan Dr.
dan teknologi.
Surisa dari Malaysia. Thank you for being here with us today. Saya juga menyampaikan apresiasi pada panitia sebesar-besarnya atas penyelenggaranya kegiatan seminar ini pada Sekretariat selaku ketua panitia, dan teman-teman lainnya yang tidak saya dapatkan sebut satu persatu pada kesempatan ini. Saya berharap melalui acara seminar ini, para peserta semua kita dapat belajar banyak hal tentang pertanian, sehingga apa yang diberikan nanti oleh para narasumber dan penyaji dapat kita ambil ilmunya. Baik, hanya itu yang dapat saya sampaikan dalam sambutan ini. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Well, thank you so much for Mr. Desmond Bernal and CMT. The next agenda is remarks from PRP of Director of Pontianak State Polytechnic that will be delivered by Vice Director 1, Mr. Insinyur Haji Irawan Sumarto, STMT. The beautiful time is yours.
The politics of speaker is just is outstanding. I also want to thank all of the listeners and all of the invited participants for connecting us meeting. And I hope you are looking forward to discussing as much as possible. Jadi saya ucapkan juga terima kasih pada keputusan dan kalian sudah mengadakan seminar internasional ini karena ini menjadi bagian dari misi politik. Yang pertama menjadi implementasi dari visi kita supaya kita mampu bersaing di tingkat nasional dan internasional. Walaupun masa organisasinya hingga tahun 2045. Jadi this is the plan, the agriculture technology department is solving its international conference in hybrid format. Therefore, it's understandable if there is a big inconvenience. Despite of that, we can look to the future of optimism. The next conference will be greater. Dengan mengucapkan bismillahirrahmanirrahim, it's now giving me great pleasure to declare this international conference open, and I wish you are great success for the conference. Saya akhiri, assalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Insinyur Haji Irawan Suharto, SCMT. Dear participants, now we invite you to take a group photo. Please move to the front to do the photo session. For the first, we invite all the participants for head of departments, ketua jurusan, kemudian juga yang dari ketua KKN, kepala KKN, dan juga all of the lecturers. Silakan untuk lebih dulu. Baru setelah itu, after that, we invite all the participants to have. So we have two sessions. So the first conversation is a high director of the department and so on. Yeah, after the session is our lecturers. Okay, say hi to Mira. One more time. Oh, our all of the lectures. You can move lecture. Okay. Mr. Sna, Tika, Mr. Mulyani. Ya, baik. Untuk semua dosen diharapkan untuk maju ke depan. Bu Sania, Bu Nancy, Bu Surya, Sultani, Rum, silakan maju ke depan untuk sesi foto bersama. Semua dosen, silakan maju ke depan. Okay, another smile. Okay, thank you for being our lecture. Thank you for the whole participants today. Please have a seat.
Thank you.
Okay, thank you so much for the conversation. Our lecture.
Sini lagi. Biar kelihatan lagi.
Well, the next of the event. Dear ladies and gentlemen, we are going to record agenda. Which plenary session invited seminar material, presentation, and discussion by four keynote speakers. This panel will be led by our exceptional moderator, Ms. Nurul Fadilah SPG LPT. I, as the master of the ceremony on this event, would like to read her curriculum vitae. Curriculum vitae. Ms. Nurul Fadilah, date of birth, Pontiac, November 5, 1982. Works experience. Vocational Universities Leaders Internship Program by Directorate General of Vocational Education, Ministry of Education, Counter Research and Technology of the Republic of Indonesia, Country University, United Kingdom, 2022. And education background. Postgraduate of Education, title specialization at University of Australia, Adelaide, South Australia, 2012. And the last is research experience. Currently applying ongoing research for 2022 Senior Australia Education Links Boards in collaboration with 13 University Australia with project advisor Dr. Julian Chen, 2022. So it's her curriculum vitae. Now, please welcome Ms. Nurul Fadilah SPG LPT to go on the stage. It's yours.
Thank you so much, Ms. Actor. Hello, everyone. How are you? It's very good. It's been nice to see you all here, all participants, incredible audience. Okay. So, we will start our keynote speakers presentations. First, we will check the audio from the speakers. Okay, we will say hi to the keynote speakers. Okay. Yeah. We will start from the first speaker, that is Professor Takuya Sugahara from Japan. Hello, Professor Takuya. Can you hear my voice clearly?
Yes, I can hear you. Can you hear me?
Okay. Hello, Professor, how do you do?
Yeah.
Okay. Yeah, it's nice to meet you. I'm here acting as the moderator for today's international conference. But first of all, I would like to meet Professor Takuya Sugahara background. His basic outlet is in Japan, February 12, 1963. And his latest education is graduate school for agriculture, Kyushu University, and his PhD degree in agriculture. Currently is director of political function research center. In 2023, the dean of the graduate school of agricultural science and Hima University. And he did really such research experience in agriculture, food science, functional animal science technology, and also did so many publications and he made work in 2017. Okay, Professor Takuya, are you ready for your presentation?
Yes, okay.
Okay, please welcome Professor Takuya Sugahara.
Okay, can you, my trial is shared. It's okay, no problem. Okay, I would like to introduce my research. At first, thank you very much for a nice introduction, Professor Nurul. I'd like to introduce myself. My name is Takuya Sugahara from Ehime University, Japan. I am a dean of the Agricultural United Graduate School of Agricultural Sciences in Ehime University. And then I'm director of the Food and Health Function Research Center. So my study focus is the evaluation of the food functions. Today, I would like to focus on the stress because the Ehime Prefecture is very famous in stress in Japan. So the stress containing many kinds of functional substances in itself. So we are evaluating the function of the stresses and developing the functional food using stress components. So at first, I would like to introduce the Japan. So this is map of Japan. So Japan have the major 4 islands: Hokkaido and Honshu and Shikoku and Kyushu. There are 4 major islands. Ehime is in one of the small island in here, Shikoku Island. So there is 4 prefectures. Then this is the Ehime prefecture is here. And here is Matsuyama. Matsuyama is the capital of Ehime prefecture. So the Ehime University is located in Matsuyama City. So the Matsuyama City is very famous and many many places to see. So this picture is the very old building, wood-made buildings. It is a Dogo hot spring. We call it Dogo Onsen. This building is very, very old, and it was built in 120 years ago, almost 120 years ago. So this is a hot spring. Yeah, there is many visitors. Too many visitors come to the hot spring. And then this is the castle, Matsuyama Castle. This also made wooden-made buildings. It's also very old, more than 400 years ago. So Matsuyama Castle is on the hilltop of Matsuyama City. So this is the view from the hilltop of the Matsuyama Castle. The population of the Matsuyama City, the 500,000 people live in Matsuyama. So the yeah, a little bit not so big city, but so there are so many buildings and many people are living in Matsuyama. So this picture shows the autumn festival, maybe October is the autumn season in Japan. So this is the Mikoshi. So the many people bring the Mikoshi to over the Matsuyama city. So very active festival in Japan. So then here's a picture of the Ehime University. This is the main gate of the Ehime University, main campus of Ehime University. This picture was taken in the spring season. So this is the full bloom of cherry blossoms. And this is the front gate picture of from the front gate of Faculty of Agriculture. Faculty of Agriculture is located in different campus. It's maybe 15 minutes from main campus. It's also in the Matsuyama city. So at the Ehime University, Faculty of Agriculture, we are developing human resources who are active in many fields. This is the citrus garden and evaluating gene manipulation and the wooden forest, something like that. So there are three departments in Faculty of Agriculture. I'm in the applied biosciences, Department of Applied Bioscience. So my study focus is the evaluation of the food function by using animal cell culture and animal experiment. So this is the strategy for evaluating the food functions. At first, we are using the animal cell culture technique to evaluate the food component function of food components. We use many kinds of animal cells, and we are evaluating the immunological function, anti-allergy function, anti-inflammation, and anti-obesity, and anti-osteoclast. This picture is the adipocytes. We are using this cell to evaluate the anti-obesity activity or non-communicable disease, like non-communicable diseases. So at first, we evaluate the food components by using animal cell culture. We put some food extracts to the cell culture media, and then we are observing the effect of food components on animal cells. Then if we can see some effect of food components on animal cells, we move to the animal experiment using mice. This is the in vivo experiment in living organisms. So we are normally using the mouse to evaluate the functions after oral administration of food stuff. So after that, we collect the serum organs to evaluate some effect of food components in mice. So we collecting some organs from the mice after oral administration. So we collected some cells from the living tissues obtained from the mice. And then we are evaluating the effect of food components in tissues by evaluating the activity of cells collected from the tissues. Tissue from experimental animals, and then cells from affected tissues are evaluated to for the function of food in animals. This we are calling it is an ex vivo experiment because we are collecting the cells we are collecting the cells from living tissue after oral administration of food components. Then we check the activity of a cell in the tissues. Then we are comparing the activity of the tissue after administration of the food stuff and administration of the just water, something like that. Just compare with the control mice. So this, I'd like to introduce the assay of animal cell culture. We are culturing many kinds of cells in the culture media, cultured in media. So we add the samples. Sample mean some extract of food. Then culture for several hours or several days. Then after that, we collect the culture media to evaluate the some markers by enzyme linked immunosorbent assay, ELISA. And then we are evaluating the effects of food components on animal cells by using the culture media. Not only culture media, but we are using cell itself to evaluate the effect on gene expression level. So we are using many kinds of animal cells. This is just a four example. This hybridoma cells, it means the kind of lymphocytes, immunological cells. And this is the adipocyte. We are using the cells to evaluate the anti-obesity activity. And then there is the human lung cancer cell line and the human colon cancer cell lines. These cells are using by the anti-cancer activity of the food components. So not only these four cell lines, but also we are using many kinds of cells to evaluate the anti-allergy, anti-inflammation, immunostimulation, anti-cancer, anti-obesity, anti-osteoclast, and something like that. So by using the animal cell culture technique and animal experiment techniques, so we are evaluating many kinds of functions of food materials. So as I mentioned, EHIM is very famous in stress. So we are evaluating the function, especially anti-allergic functions of components contained in stress. So in this presentation, I would like to focus on nobiletin. The nobiletin is a kind of polymethoxyflavonoids, polymethoxylated flavon. It had the anti-inflammation, anti-tumor effects in vitro and in vivo. The nobiletin was also found to potentially inhibit cartilage degradations. So in this presentation, I would like to focus on the anti-allergy effect of nobiletin, because the allergy is very, very serious problem in Japan. Maybe in Indonesia, there are so many people suffering from the allergy, like dermatitis, atopic diseases. So this is a type 1 allergy. There is 4 kinds of allergy symptoms. Pollinosis is very serious problem. Maybe 1/3 of Japanese people suffer from the pollinosis. The pollinosis is the major allergen for Japanese people. So the pollinosis is categorized in the type 1 allergy. Type 1 allergy is this slide shows the mechanism of the type 1 allergy. The mast cells and the basophils kind of white blood cells participate in the type 1 allergy. Generally, the IgE receptor on mast cells and the basophils. So the IgE produced by the lymphocytes bind to the basophils mast cells via IgE receptors. IgE, one of the reason of the allergy, type 1 allergy. IgE is the very important immunological proteins induce the allergy. So IgE is produced by the lymphocytes, and then the IgE in blood is bind to the IgE receptors. So the IgE recognize the allergen. So the the pollen bound to the IgA, bind to the mast cells or basophils via IgE. So the binding of allergen to the IgE on mast cells induce the degranulation. This means the release of histamine and leukotrienes. This means the degranulation. So basophils and mast cells have the granules inside of the cells. The allergen induction induce the release of granules from cytosol to outside of the cytosol. This means the degranulation. So the granules, there is a histamine and leukotriene in granules stimulate the some epithelial cells, and then induce some symptoms of the type 1 allergy. So to prevent the type 1 allergy symptoms, decrease or inhibit the degranulation is very important strategy for reduce the type 1 allergy symptom. So we are using the rat basophil cell line RBL2HC cells to evaluate the anti-degranulation activity of noveretin. So noveretin, as shown in this figure, noveretin suppress the degranulation of rat basophil cell line induced by antigen. Antigen-induced degranulation was dose-dependently inhibited by the addition of noveretin to the culture media. This means the noveretin have the anti-degranulation activity. This means the noveretin can inhibit the release of histamine. Histamine induce the allergic symptoms. So this is very important point. Noveretin can reduce the histamine release from basophils. So we are evaluating the mechanism of the noveretin. This is the IgE receptor and IgE. Here is the binding of antigen to the basophils via IgE receptors. So the binding of antigen to the IgE induce the signal transduction inside of the cells. So the RIN-seq, PS3 kinase, and the PLC gamma 1 or 2 sequentially activated. And then intercellular calcium ion concentration is increased by this signaling. So the increase in the calcium ion concentration induced the release of the granules. This means the degranulations. So the histamine stimulate the some epithelial cells. And there is one signaling pathways. However, there is another pathways. This pathways, another pathway is FIN PI3 kinase AKT pathways. So in this pathways stimulated by the antigen finally induce the microtubule formation. So the formation of microtubule induce the granule release. So mean the degenerations. So there is two pathways. One pathway is the calcium ion dependent pathways. Rin-seq, PI3 kinase, PI3 gamma 1, 2, and the calcium ion concentration increase, and then release of the granules. And the other pathway, this blue line pathway is the calcium ion independent pathways. This calcium ion independent pathways induce the microtubule formation, stimulate the microtubule formation, and induce the degeneration. So there are two pathways, and then we are evaluating which point nobrating can inhibit the sequential activation of degeneration signaling. So as a result of the Western blotting analysis, nobrating can suppress the activation of PI3 kinase and activation of AKT, as shown in these figures. Will clearly suppress the PI3 kinase activation. AKT is a downstream of the PI3 kinase, so the inhibition of the inhibited activation of the PI3 kinase is resulted in the inactivation of the AKT. So the point of the inhibition point of nobrating is the activation of the PI3 kinase, like wortmannin. Wortmannin is the anti-allergy medicines. So as well as anti-allergy medicine, nobrating can clearly suppress the activation of PI3 kinase. So the nobrating is suppress this point. Then downstream of the is also suppressed by suppressing by PI3 kinase activation. So this shows the effect of nobrating in mice. So this is the PCA reaction in the cells. IgE injected in the ear, and then after that antigen injected from the tail vein. Then the antigen reach to the ear and then bind to the IgE. So the vessel, blood vessel, the permeability of the blood vessel is increased, and then blue dye leaked from the ear, and then the control mice, the ears of control mice are colored and stained in blue. However, intake of nobrating clearly suppress the leak of the blue dye. This means the suppress the increase in permeability of blood vessels. This means the suppress the degeneration in ear. So the intake of the nobrating can easily clearly suppress the degeneration in mice. So intake of nobrating is effectively suppress the degeneration in body. So this is the we'd like to introduce the synergistic effect of nobrating and beta-lactoglobin. Nobrating is the functional component in stress. However, nobrating itself also have the suppressing activity. However, effective suppression, we are searching for some substances synergistically suppress the degradation with nobility. So beta-lactoglobin is the major whey protein contained in the cow milk. And then it also have the anti-degradation activity from by our experiment. Our experiment indicated that the beta-lactoglobin also suppress the degradation as well as nobility. As shown in this figure, the beta-lactoglobin also dose-dependent suppress the degradation. So we mixed with the nobility and beta-lactoglobin to evaluate the anti-degradation activity. Only nobility and the only beta-lactoglobin have the little bit suppress the degradation. However, a combination of nobility and beta-lactoglobin significantly suppress the degradation of RBL2H cells, animal cells. So this slide shows there is some concurrent and synergistic effect on anti-degradation, anti-allergy. So we are moved to the animal experiment. So the stress-peel powder and yogurt containing a beta-lactoglobin, and the stress-peel powder containing the nobility, and the mixture of stress-peel powder and the yogurt. So we are using the pseudoponosis model mice. So the CRY-J1 is a pseudoporin allergen. So injected in the nasal anesthesia for 3 times, and then after that we injected small volume of the CRY-J1, and then this one week these samples are or administered in mice, ponosis mice. So as a result of the 1 week in oral administration, stress-peel and yogurt and stress-peel plus yogurt, stress-peel with yogurt clearly suppress these symptoms, allergy symptoms. So as you can see in this slide, combination of stress-peel and yogurt effectively suppress the ponosis symptoms in mice. And why combination of the nobility and the beta-lactoglobin is effective for anti-degradation? So we are evaluating the mode of action of these 2 substances. As I mentioned before, nobility suppress PI3 kinase activation and then decrease the degradation. On the other hand, beta-lactoglobin suppress the SYK activation. So beta-lactoglobin and the nobility suppress the different signaling proteins, and then the nobility suppress the PI3 kinase activation. Beta-lactoglobin suppress the SYK activation. So these 2 factors can suppress the different point of the signal transduction. So we can see the synergistic effect of nobility and beta-lactoglobin to suppress the degradation. So after that, we are moving to the human trial. So the ponosis symptoms can be decreased by intake of stress-peel containing yogurt. This is the before yogurt intake. Yogurt means the stress-peel containing yogurt. Before yogurt intake, there is some allergy symptoms in eyes. However, after yogurt intake for 2 weeks, completely suppress the ear symptoms, as shown in this figure. So the intake of the stress-peel yogurt can suppress human ponosis symptoms. So after that, we are developing some products containing Straspear containing yogurt. This is the final product of the Straspear yogurt. We are developing drink yogurt with milk company in Ehime Prefecture, and Ehime University students participate in the product development. So we are successful to the what happened? Okay. This is the regulation, functional food regulation in Japan. So there is the 3 types of functional food in Japan, government controlled. So the recently food with function claims, FFC, is the newly established functional food regulation. So many of the items developed in these 8 years. So we plan to be FFC. We are planning the Straspear yogurt to be a FFC. So we are finally develop modify the Straspear yogurt, and then we apply for the FFC. So finally, last year, we can successfully enroll the Straspear yogurt as FFC. This is a health claim. This product contains the nobility and beta-lactoglobin. Stimultaneous intake of nobility and beta-lactoglobin has been reported to produce eye discomfort caused by the pollen, dust, house dust, and so on. So we can successfully enroll this product as a FFC. FFC is a government-regulated functional food. So we can develop the FFC by using Straspear and then yogurt. So not only this yogurt, but also we are developing many kinds of products using Stras. This is a tablet, and this is the tea using Stras leaf. And then there's a tablet and the jelly and some kind of products developed by using the Straspear. Okay, thank you very much for your kind attention. So in my presentation, hopefully you are interested in the function of Straspear. And not only Straspear, but also we are evaluating some spices, the function of spices by collaboration with Indonesian universities. So the spices are also very important products in Indonesia. Clove and cinnamons also have the many kinds of functions. So we are evaluating the function of spices and herbs. So I would like to collaborate with the evaluation of the function of spices with Indonesian Institute. Yeah, thank you for your kind attention. Thank you so much.
Okay. Okay, terima kasih, Professor. Thank you for your
Thank you.
wonderful explanations. Okay, give applause for Professor. Okay, our respectful audience, we will open the discussions after finish all of the keynote speakers' presentations. Okay, next session is the presentation from Dr. Samsul Haq from Charles Sturt University, Australia. Hello, Dr. Samsul. Can you hear my voice clearly? Hello? Dr. Samson? Are you with me?
Yeah, now it's better.
Okay. Hello, Doctor. How do you do?
Yeah, very good. Fantastic. How are you?
Okay, I'm good. Alhamdulillah. Thank you so much for your time. So, before that, I will read Dr. Samson have curriculum vitae. Dr. Samson is currently a lecturer in soil science at Charles University of Australia. He conducted his PhD at CSU on factors affecting the oceans across capital, performing floating price in southern New South Wales. He also examined the soil-related barriers in early drought subdivision, following price and mitigation strategies by optimizing economic practices. He has worked in Bangladesh in soil resource management and soil agronomy and crop science courses at BCRR Agricultural University. He also served as an agricultural extension officer for natural resource management at UNDP Bangladesh. And Dr. Samson currently teaches soil science, soil management, summer cropping system subjects at CSU Australia. As a senior research scientist, Dr. Samson has been responsible for the European Soil Program for 2 years, managing hundreds of field trials in World Agri and agricultural crops. As a researcher at CSU, Dr. Samson also participated in GRDC, funded long-term crop rotational trials to investigate soil health and with seed banks persistent, along with various industry-funded research trials and MLA-funded soil down detailed relations on the soil function project. Dr. Samson was the recipient of the SUACR Research Award studying the impact of soil management on soil fertility and nutrient cycling. And currently, Dr. Samson is working on a soil extension smart farm project entitled Building Resilient Soil in the Riverina, funded by DAFF and other projects studying irrigated crop productions in the riverina and use the solar panel systems for safe production of dryland pastures. We also published more than 30 research articles, conference proceedings, and grower notes. Wow, that's sophisticated professional backgrounds, doctor. Okay. Well, let us have Dr. Samson for his presentations. Please, Dr. Samson, the time is yours.
Okay, thank you very much, Professor. So I want to share my screen and want to see that if you can see it. This is good. Yeah, okay, cool. So just want to see. Okay, so today I want to talk about biochar and its importance in our cropping system. Thanks, Professor, for giving me a very good introduction. Yeah, I think you say everything about me. I am Shamsul, came from Bangladesh in Australia, did my PhD, postdoc, and now lecturing in soil science subject. Now, I really like the topic of this conference, and one of the sentences actually strike me: the future of agriculture, exploring emerging technologies, scientific breakthrough, and sustainable practices. Now, it's really important to carry on our sustainable practices. So we did really good improvement in agriculture. We are also improving, but is it sustainable or not? So that's the big question. Now, I am now in Australia. So you can look at, I'm here. The town name is Wagga Wagga. And this is around 4.5 half hour drive from Sydney, same distance from Melbourne, 2.5 hour from Canberra, and I think you are here. So we got multiple campuses, but before going ahead, I want to acknowledge the country, the indigenous people in Australia whose land we are sitting at the moment, and their past and present generations. Now, I am working at Charles Sturt University, and within the map I showed you that we got multiple campuses, and we are one of the largest inland university in Australia. We got a really diverse discipline, but the campus I am sitting at the moment is really rich in agriculture. So if you think about agricultural science, Charles Sturt University Wagga campus is the main one. Totally, we got around 43,000 enrollment, undergraduate, postgraduate, and PhD. I think I checked the other day, it's around 110 countries, different countries, students coming here. Now, today, I want to talk about biochar, but before going ahead, I want to tell something about soil carbon. So when you look at the big picture of carbon cycle, and if I tell that this is dark black color, which is storage gigaton of carbon, and the blue color is at the fluxes of carbon. And you can see that all of our activities, including fossil fuels, chemical production, our agricultural production, our vegetable, all actually impacting soil carbon. Now, this is the above ground activities. But if you think about what happened in the ocean, also you can see that there is a bit of activities going on here as well. Soil is a harbor of a huge amount of carbon, and it's impacted by our cropping activity as well. So the forest plan and our cropping, including our pasture and agricultural activity, did lots of fluxes in soil carbon. Now, what we can do, we can stop. Like we can say that, oh, okay, this is something we can't do anything, then we can stop our activity. But the other thing we can do that everyone, I can see you on the screen and who are presenting here. I think if we can start fighting with our own position and try to achieve something within the small scale, then as a whole, we can achieve a healthier soil and healthier ecosystem. Now, I am, soil cropping. So my interest in this area where the tractor is, so what we can do it here. So if you think about the factors, so you got crops, we got carbon dioxide going to the atmosphere by respiration. We are using carbon dioxide photosynthesis. Microbes are also here, and they are using the carbon dioxide as well for their respiration. So all of those factors actually indicating that how total carbon will be stored here. Now, one of the key player is microorganism. Now, it's a big subject, but if I can narrow it down, that what happened with the microorganisms. So if we increase the global temperature, or if somehow we are acidify our soil, acidification happen for many ways. So for example, carbon dioxide is coming to the soil and then it's react with water and forming soil and acidify the soil. So either way is not good environment for the microbes. So they don't feel good. So they need neutral type of pH. They need ambient type of temperature. But if you change that one, then microbes are not comfortable staying here. When they are not comfortable, then they are thinking, okay, I might leave that place, or they might die. So the microbes is the important factor for our cropping system, our nutrient cycling. So if there's no microbes, then we got the problem. So you can stop here with the microbes part. So now we know that we really need microbes. Now, if we come to that point here, that we got our agricultural production, we want more food, that what are the options? So for the cropping, we actually apply lots of fertilizer. Now let's look at here in Australian situation. You can see that the fertilizer price started increasing here, and you can see that it just reached the peak here. We know that we got global unrest and different areas in the world has unrest condition, but the truth is the price is increasing. Once the price is increasing, if we think about the right hand side graph, that the relation between fertilizer and wheat crop, you can see that the ratio is high. That means you need to apply more fertilizer, that means more money, spending more money to get the same amount of crop. So the production cost is also increasing. So that is the one side of the coin, that we are actually in a position that we need to spend more money or more fertilizer to get the crop out. Now we got few consequences. Definitely got environmental consequences. You know that the fertilizer we got 30 to 50%, sometimes 60% efficiency. So that means the remaining actually fertilizer is remain in the soil, in the atmosphere. So it actually degrading our cropping environment, the whole ecosystem. But I want to point out the other point, that the microbial community. So there is a research, Rain et al. If you want to read that paper, you can see that the long-term nitrogen application is negatively impacting bacterial community. Because it's negatively impacting bacterial community, that means we need to think about what we can do. Now, one question: should we stop applying fertilizer?
Oh yeah.
The short answer is no. We can't just say that stop applying fertilizer. We are in a position globally that we need a set amount of crop to feed the world population. So that means we just overnight cannot tell that, okay, stop using fertilizer. But what we can do, that we can creating some environment that we can, the system is sustainable. Now, one of the sustainable thing we thinking is using beneficial microorganism. So the beneficial microorganism, I'll come in a minute that what are the beneficial, but simply that the in soil system, for example, from conversion from organic nitrogen to inorganic, we need microbes. So that means they are beneficial. Transforming some other nutrients, also microbes actually doing the reaction, so they are beneficial. There's some harm.
Oh putus nih. Dari sananya ya kayaknya. Kalau kita internet.
Dr. Sasha? There are any technical problems there?
Putus ya. Waduh. Dari sananya.
Okay here.
Hello?
It's okay. Maybe there is a technical problem in Australia
Oh, okay.
from Samsul Haq.
Okay, I think we can wait for 3 to 5 minutes, waiting for the connections with Dr. Samsu. Dari sananya.
Dari sananya. Mister Tako ini. Dia masih.
Bisa. Bapaknya juga.
Sorry, can you see me now?
Okay.
Oh, I don't know what I somehow,
Okay, finally.
yeah, I think Zoom doesn't like me. Say that or get out. Okay,
Alright.
I can share my screen. Yeah, sorry about that, but can you see me now? Okay, cool. So what happened? We got biochar, which is I can start from here. So the biochar is a so the biochar is a pyrolyzed plant material. So if we think about, we use high temperature, but in absence of oxygen, then we can get the stable carbon. Now, it says in the literature that the phosphorus can increase 4 to 6 times. Tissue heavy metal can decrease 17 to 39 times, and it can build organic carbon minus 21 to plus 20%. So this is the point you need to remember. That is a minus and plus as well. So that means if you don't use judicial use of biochar, that it could be negative. And we can say that the non-carbon dioxide greenhouse gas emission is also reduced by 12 to 50%. Now, what we are trying to do is set a model. Then we are thinking that if we add biochar and if we add microbes like the biofertilizer, and if we combine them, what happened to the soil activities, and is this impacting on the plant growth? So for the plant, we pick wheat crop. Now, what we are trying to do, that we set a few treatments, and we use some beneficial bacteria, some mycorrhizal fungi, only biochar, untreated control. We put no plants, and we use synthetic fertilizer as a control. We use 2 different type of soil. If you know Australian soil, then we got to red chromosol. So the chromosol is a duplex soil. You can see that we got a distinct layer here. So we got 2 different type of soil texture here. We called chromosol. It is one of our common soil. And the other one I use gray Vartosol. So Vartosol is a cracking clay. So you got all the way through soil with the carbon blend in here. Now we use the randomized complete block and wheat crop, and we take some data on plant biomass and microbial population and community. Now, so these 2 planted microbial treatment, one is Bacillus, another is mycorrhizal blend. So the Bacillus is actively, we got Bacillus species, and we got mycorrhizal blend, we got Endomycorrhiza, Ectomycorrhiza, some Trichoderma, and some other microbes as well. We got two different soils, and we used the Bacillus mycorrhizae and biochar, and we did some test of all of the thing. So I'm not going in that detail, but at least we can see that the carbon component of this product is really, really high. So what we want, the theory is, if we provide a carbon rich environment, then does those microbes feel better? Are they healthy? Are they grow well? So that is the objective of that one. Now for the microbes, we need aeration, we need soil moisture as well. Now if we can provide biochar, and if we can, and at the same time, if we can add biochar and the microbes, then what could happen, that the microbes might get better environment. So that is our hypothesis. So let's see what we got. So we use the crop total dry matter and crop harvest, and this is the two different soils, and we got 8 different treatments. So throughout my presentation, I want to say Bacillus plus biochar, Bacillus minus biochar, mycorrhiza plus biochar and minus biochar. We got biochar only, untreated control, no plants, and just fertilizer. So with the fertilizer, you can see that definitely we got better crop dry matter compared to the others. But when you look at the crop harvest index, which is the ratio between above ground head of the harvest particle of the crop and the biomass, and you can see that the ratio is actually lower compared to the treatment, for example, Bacillus plus biochar. What does it mean? This means that even we are getting less crop diameter, still the partitioning is good compared to the fertilized treatment. So that's really important, that if we can use that product judicially, we might get better crops. So that is the other thing, that how we can improve this dry matter. That is a separate thing. Now, plant carbon, nitrogen, and CN ratio. So this is the total carbon and nitrogen, and this is the ratio of carbon and nitrogen. And you can see that the CN ratio is really low in here, but here we can get around 35 to 45, something like that. So our benchmark is around 25 or roughly like that. So some of the treatment, when you are not, when we are using Bartosol, you can see that we are getting here, when you put the Bacillus, we got higher CN ratio. So higher CN ratio means that you are going towards immobilization. So depending on the factor, it might be good or bad for your cropping. Ammonium nitrogen, yeah, so we got really high ammonium nitrogen here, and we did not find significant difference among the nitrogen partitioning. So this is probably due to the very short duration of one season crop. Soil pH, and we found that the soil pH is not changed in two different soil, but when you compare the biochar adding or without adding, we can see that the pH is changing. So that's really important for our Australian soil, where we are getting really acidic soil, and we want to adjust the pH. So that could be the organic way of adjusting soil pH. Soil phosphorus is definitely, there is a difference phosphorus between Chromosol and Bartosol. You know that the Bartosol is mostly our rice growing soil here, and when it's flooded, the sesquioxide actually, the ion attached to that one break down, and then phosphorus release, so we got more phosphorus here. But here the important thing is that when we add biochar, and compared to the without biochar, you can see that we are getting more phosphorus here compared to that one. So that means this might be useful strategy to release phosphorus as well. Now let's come to the soil bacterial community, what we can get it. So this figure is actually two different so the same treatment, gray vertisol and red chromosol. So the same treatment, two different type of soil. So this angle actually showing you the difference between the soil, but it's difficult to interpret from that one. So what we did another type of analysis, that where we got all of the treatments. We got red chromosol and gray vertisol, and we all got all of the bacterial community. Now this specification of or identification of all of the nutrients, earlier it was difficult, but all of the species now very easy. If you want to do next generation sequencing, send the sample in the lab and you can get really good data set from that one. So if you blow that one and present it in the other way, you can see that particularly this one, you can see that really dark color. So this is a heat map. So with the heat map, you can see that there is a sharp difference between two different soil. That's normal. But within the soil, you can see that some of the species is more compared to the other. So when it takes actually days to discuss about which species do what, but I just pick one for today's discussion. So these fibrobacteria, you can find that the treatment where we is red chromosol, and when we put mycorrhiza plus biochar, we are getting really high number. So that means if we use that treatment, we can get this particular bacterial species abundant in number. What does it mean? So we can then search what are the function of these bacteria. So then if we go some literature, and we can find that some of the cellulose degrading bacteria, those are these bacteria, these fibrobacteria, they can dissolve this cellulose driven carbon retention. Now we got something that we can isolate on a type of bacteria, and we can now work in the field and see that what the practical use of that one. So from there we can go forward, and I can give one of the example. Thanks Jaini for giving this figure. So here this we are thinking about oil palm empty fruit branches, and then we use these as a source of biochar, and we make nano biofertilizer, biochar based fertilizer. And we want to see that if we mix any microbes, for example, that one that we just found it, and if we add it in rice paddy with that fertilizer and without fertilizer, what could be happen? Our hypothesis is that if this is degrading the cellulose, so the rice stubble stay there for longer, and in 5-6 years is never break. So if that microbes can break that cellulose, then gives good soil structure, and eventually you are getting very good crop. So we actually thinking on that point that, okay, we got some bacillus or microbial fungi, and we can isolate some, and with that one we can actually make some commercially sustainable fertilizer which can be used as a cross specific. So for example, in a rice crop or a paddy with lots of cellulose based materials, so we can actually use that one and use this as a fertilizer. Yes, I can stop here. Sorry for the interruption, but I got funding from Charles Sturt University, and this is my research team. I'm really happy to collaborate with any of the people. And I can see it in the screen, and this is my contact address. You can contact me. Any question? I cannot hear you actually. You are muted. I cannot hear you.
the papers.
Yeah, I can hear you now.
Oh, masih mute. Mana? Lagi. Masih. Sebentar, siapa? Siapa yang?
Hello?
Sebentar ya.
Hello. Yeah, I can hear you.
Okay, sorry for the technical things. So, please be close for, that's how we will open the discussion after all the speakers, present the papers.
Okay.
Okay, thank you so much. Now
Okay,
let's
thank you.
move on to the next sessions, the presentation from Dr. Hafiz Shohai Mazhab from the University Institute of Science in Emergency Medicine, Guangdong Academy of Medical Science, Guangdong Provincial People's Hospital, Guangzhou, China. Hello, Dr. Hafiz, can you hear me?
Hello everyone.
Hello, how do you do, Doctor?
Alhamdulillah. Assalamu alaikum everyone.
Walaikum salam warahmatullahi wabarakatuh. Thank you so much for your time. So before that, I will read your curriculum detail. Dr. Hafiz Shohai Mazhab is currently a postdoctoral researcher in Institutes of Science in Emergency Medicine, Guangdong Academy of Medical Science, Guangdong Provincial People's Hospital, Guangzhou, China. He got his PhD in environmental microbiology at Fujian Agriculture and Forestry University, Fuzhou, China, in 2020. And the title of the dissertation is the effect of co-selections of antibiotic resistance to heavy metals resistant genes and genome plasticity of bacterial adaptations to high levels of contaminants in poultry, published in Level 1 journal and publication Level 2 journals. He did also many publications, both national and international, with research interest in the antimicrobial resistance, or AMR, that how resistance genes interact and represent heavy metals and microbiomes with experimental and computational approach, along with evolutionary study of microbial genome using bioinformatics tools. He also working on relationship between above-mentioned determinants with acute allergic conditions and relate them to contribute toward the states of anaphylactic shock in emergency medicine. He also involved in international conference presentations internationally. He also published publications and received many awards, international awards. Okay. Our respected audience, please welcome Dr. Hafiz Shohai Mazhab. This time is yours.
Thank you so much for inviting me here in this great session. Can you see my slides here?
Yes, we can. Yes, we can see.
Okay.
Yes, doctor. We can see your slides.
Okay. Thank you so much for inviting me here for the presentation. And this is a very unique topic to discuss about the sustainability in agriculture. And you know, the agriculture is a very vast term. It's not only depending upon the agricultural soil and crops, but it also has a major contributor, which is a livestock. So being a veterinarian, being a veterinary doctor, my major concern in agriculture is livestock. So today I will talk about the sector of livestock and the problem and the serious problem which are arising in this sector, and they are being addressed on many great forums, and the studies are going on to cope up the problems in this agricultural sector of livestock. Here today I will discuss about the antimicrobial resistance and the role of antimicrobial resistance in the presence of heavy metals. And this is a unique topic, you can say, that it is very less studied, that heavy metals, they are interacting with antibiotic, and along with antibiotics, metals, they are increasing the resistance in the microorganisms. So this is a kind of a very unique thing, and that we have done recently. And this is just a general idea for the antibiotics, and you know that antibiotic resistance is going to increase day by day, and especially it is coming from our livestock sector, because we are giving lavishly the antibiotics to our livestock, whether it is poultry, whether it is large animals, whether it is small animals. We are giving them lavishly, excessively, to get more and more production. And but unfortunately, the consequence is the resistance is coming towards our plates and coming towards the human being, and then is going on to the environment, and then the cycle goes on. This was the just an introduction, and if you see, the antibiotics, they are used not only for the disease prevention, but only for getting more growth, and to enhance the animal growth rate and the productivity, not only the infection in livestock sector. Because but in human being, you see, if we are sick, we are taking antibiotics. If we are not sick, we are not taking antibiotics. But in addition, we are taking food from the animal side, but that food is containing so many antibiotic residues which are coming from the livestock. So this is a very growing concern for one health issue, because one health issues are those issues which are for both human being and for the animals. So these are kind of combined kind of issues which should be addressed immediately. So coming towards the antibiotics, they are excessively used, and they because they are excessively used, so their antibiotic resistant genes, they are excessively present in the environment, and they are now considered as global environmental pollution. They are considered pollutant. These resistance genes, they are considered as pollutants now worldwide. They are considered now, and US have studied that each year there is 2 million morbidities. Morbidities means 2 million diseases appear every year, and 23,000 deaths happen every year due to this resistance. Because if we are sick, we take medicine, but those medicines cannot affect on our body. We cannot recover from those medicines because we are getting very resistant towards those antibiotics. That's why we are getting more and more sick, and we are getting diseases and sometimes deaths. So now, what is the growing concern, and the topic which I want to address today, and the things that we have researched recently, is that the relationship of antimicrobial resistance with heavy metals. So this relationship is very new, although it is very old, but the discovery of this relationship is very new and very unique. As I already have discussed, that we use a lot of antibiotics, but I have not discussed that we also use a lot of heavy metals. Heavy metals, how we use for the livestock and for the human being in both ways. We are using heavy metals a lot in case of supplements, in case of boosters, in case of immunomodulators, just like food supplements, or just like we say we are taking multivitamins, or we are giving animals to the multivitamins or the boosters or the feed efficient drugs. All those things, they contain heavy metals for the benefit of the body, although they are going inside the body for the benefits, but in one way. But the other way, they are inducing the resistance in our body. So I will discuss it later, that how they are inducing the resistance in our body. And the important thing is both antibiotics and metals, they are poorly absorbed. If animal is given antibiotic, if animal is given immunomodulator, they go inside the body and they are not properly absorbed, and then eventually they comes out of the body and become the part of environment. So the resistant genes stays there in the environment, and through the cropping they come again to the animal body, and this cycle goes on again and again. Now, our hypothesis for the study, because during my PhD we try to cover up this topic, and then we have hypothesized that there must be a link between antibiotics and heavy metals for the co-selection, because co-selection is a thing where if one thing is not present but the other thing is present, so the other thing can play the part for the first thing. So it means if antibiotics is not present but the heavy metal is present, so heavy metal will play a role for the selection of antibiotic resistant genes. Means it means that heavy metals are playing the same role as antibiotics for the increase in resistance. So to prove this hypothesis, we started a study and we made a plan for the project. Here we took some samples from chicken. We went to the farmhouses, chicken farmhouses. We found some samples. We did antibiotic analysis for the concentration analysis and heavy metals analysis, and then we did some DNA extraction for the molecular studies and further things for the completion of the project. And the major technologies that we used in our study are like ICP-MS, LC-MS for the antibiotic concentration analysis, and the SPARChip QRT-PCR, and this is very thing which I will discuss later, and then Illumina sequencing for the 16ers. And smart chip technology. Sorry?
We have the problem to see the slides. The slide is not moved yet.
Can you please come again?
The slides. The slides are moved. The PowerPoints or the slides.
Is okay now?
Yes.
Yes, we can.
Yeah. Thank you so much. Sorry, no.
Okay, thank you. This is smart chip technology. I think most of the students and professors are here and they know very well about the qPCR and RT-PCR and all the PCR technologies. And this is one of the qPCR technology, but this is a high throughput qPCR. And this is smart chip. Here you can see the chip over here. In this chip, we can assess the genes, almost or more than 300 genes we can assess through single chip. we can apply the assays, more than 300 assays, with more than 300 primers, and we take a result in a single go with more than 300 primers, and we can get the analysis of that results very efficient way. And if it comes towards the analysis, for the analysis, we first we validated a method in which we used different kinds of matrices in one go, using one method, like chicken manure, feces, feed, and sediment and soil. All these things can be assessed using one method, and we assessed these almost 40 antibiotics in one go. Can you hear me?
Yes, we can hear.
Yes, doctor, we can hear you. Thank you.
Okay. So this is just the initial step to start the project, and then uh, we got some results, the initial results for the distribution of antibiotic resistant genes, mobile genetic elements, heavy metals, and antibiotics. So in this graph, this is a generalized graph. In graph A, the distribution of uh, antibiotics in three types of samples. We took feed sample, we took feces, which we can call manure, and then then we took soil samples, and this is distribution of antibiotics inside. And in group D, you see the concentration of antibiotics that we found in our analysis, that tetracycline is very high in our results, and afterwards is the fluoroquinolones. Tetracycline is very excessively used, even it is very resistant now, because tetracycline is not working in the field, because there is excessive use of this drug, and now it becomes very resistant, so it's not working. And if we see in the graph C, for the metal concentration, we found that very high concentration of zinc was found inside the bodies of the animals. And then we got the graph for antibiotic resistant genes. So these are the resistant genes. Different genes are expressing very high, and some genes are very low. And we also found some mobile genetic elements, which are transposons and integrases. They are also very high, because they are very important for the horizontal gene transfer. And is the enrichment graph. We can see the heat map. In the heat map, there is a red area, and this red area is showing that all these samples belong to the feces samples, and these feces samples are very rich in the resistant genes. If you see the other samples like feed sample and soil sample, in the right side, they are not very rich, but the manure samples which is coming, manure feces which are coming from the body, they are highly resistant for the resistant genes. And if we talk about the diversity, microbial diversity, the microbiota inside the body, if you see, not only the inside the body but also in the environment, because this study was both, we compared both parameters from inside the body and from the environment also. So if you see the soil, the graph, soil is very diverse in microbiota, and then comes the feces, and then comes the feed, because feed was having almost none, the diversity of microbiome, but soil is very diverse in microbiome. And then here is the final and very important thing that we found after analysis, that there is a co-occurrence between antibiotics and and antibiotic resistant genes. If you see these nodes, arsenic, copper, zinc, cadmium, and lead, they are the metals. And on the other hand, you will see quinolones, tetracycline, and fluoroquinolones and other things. They are also present, but the clusters which are formed with metals, along with which are relating towards the antibiotic resistant genes. So all these all these branches, they are going towards the antibiotic resistant genes, but the main nodes, they are towards the metals. If you see, the metals are more prominent and they are co-selecting all these antibiotic resistant genes, which shows that the metals are also very important in the co-selection of antibiotic resistant genes in our environment, because we don't care that we are taking metals or how the metals are interacting with environment, how the metals are going towards the animal body, and how much excessively we are giving metals to boost their feed intake, to boost their growth, to boost their production. We don't care mostly. But on the other hand, these metals, they are affecting adversely on the resistant genes. So the animals are getting more and more resistant, and the drugs, if in case they are sick, we have to give more stronger drugs to cover the disease, because they are getting resistant. And that's why this is called a pollution of the environment. Here is a graph that we found a link from microbiome and resistome, and we found a very beautiful picture from the pollutes analysis. If you see the nodes here, if you see these nodes, they are all gray, and the gray pretends to microbiome, and all the other colors, they pretend to the resistomes, the antibiotic resistant genes. So means these microbiomes and resistomes, they are interlinked to each other. As a summary of the whole project, if you can see, both livestock and human being, they are affectees of these environmental pollutants. So we have to control the use of the antibiotics, we have to control the use of metals, so that we can be safe and we make our environment healthy. And if you see, it was the whole project, and we took the sample and did the analysis, and we found very good results. And we found a very beautiful picture of co-selection of metals. If you see here, the metals are putting a pressure of 5.7%, while antibiotics are not putting a pressure, co-selection pressure for the resistance. So metals are more important for co-selection of resistant genes. As a conclusion, we can say that bacterial composition and horizontal gene transfer are major influencing factors on the composition of antimicrobial resistance. And this is a very well-known thing, that microbes are very important in the whole universe, not only the environment, in the whole universe. They are very important. Along with microbiome, metals are the major influencer in the antibiotic resistant profile, and they are playing a good role. And so we have to have a serious consideration which is required to mitigate the risk of this metal induced co-selection. So we have to control our use of boosters or supplements, to the livestock so that we can have a healthy and sustainable agriculture, and that sustainable agriculture will come towards our human beings. So we have to be very careful regarding the use of metals. So this was our theme from a PhD, and thank you so much. In case you have any questions in future, you can contact me, or you can ask me anything now. I'm available. Thank you so much.
Then I'm happy for your presentation. Okay, now this last but not least, we have our last speaker, Dr. Sulisa Morkile. Hello, Dr. Sulisa. Can you hear me?
Yes, I'm hearing
Hello.
you. Oh, hi.
How do you do that?
Hi, I can hear you, yes.
Okay, thank you very much. Okay, please allow me to read your curriculum vitae first. Dr. Sulisa Morkile is a lecturer in agrotechnology program, Faculty of Resource Science and Technology, University Malaysia Sarawak, UNIMAS. He got his PhD in post-harvest technology, division from post-harvest technology, schools of bioresources and technology, King Morkus University of Technology, Honolulu in 2020. Now she is a postgraduate student in Diploma in Higher Education, Teaching and Learning, Center for Applied Learning and Multimedia, University Malaysia Sarawak, since 2022. She also wins awards and scholarship internationally. One of them is Best Video Presentation for Smart Uni IoT Hackathon 2023 in Malaysia Tech Week. She also wins Smart Fertilization and Utilization System by MDEC through collaboration with IATA Samarang or Digital Farmer in 2023. She also got postdoctoral research fellowships in 2020 and 2021. Now she is involved in Malaysian Society of Plant Physiology Professional Membership and International Society of Horticultural Science or ISHS. She also did her research grant and international publications, and many research experience with both nationals and internationals. So, okay, now please welcome our Dr. Sulisa Morkile for this presentation. Please welcome. The time is yours.
Thank you. Thank you, Madam Chairman. Let me share my screen. Okay, can everybody see my slide now?
Yes, we can see. Thank you.
Okay, all right. Okay, all right. Thank you. Thank you for the kind introduction, Madam Chairperson. As you aware, my name is Sulisa Morkile. I'm currently teaching in the Agro-Technology program in the Faculty of Resource Science and Technology, University Malaysia Sarawak. I think people from Montana actually know well where is UNIMAS, so I just do a simple introduction. So where are we located? UNIMAS, we actually located in Kota Samarahan, which is, I think we are neighbors, right? So very near to each other. So it is UNIMAS, and then currently I'm teaching Agro-Technology program. Just for your information, this program is a new program established in 2022. We have second batch now. Already finished the first batch, not yet finished. Now this is second batch, 2023. So somewhat I'll be teaching in Agro-Technology program. We have like urban agricultural technology, plant physiology, smart agriculture, science. I think is quite common. And you are welcome to join us if you are interested. Okay, all right. Today I would like to talk about chilling injury in horticultural crops. I'm not sure if anybody is familiar with chilling injury. So what is chilling injury? So chilling injury is defined as damage to the chilling sensitive plant species when they exposed to low but non-freezing temperature. So what are the sensitive plant species? They are those mostly from tropical and subtropical, because we actually grow the crops in hot climate. So if you store them in cold climate, they're gonna like having some sort of like cold shock. Okay, so for the tropical and subtropical origin, they can only tolerate the minimum 10 degree for the cold storage, and for the temperate crops, they can tolerate up to as low as 5 degrees Celsius. Okay, why are we talking about chilling injury? When we talk about chilling injury, when we talk about post-harvest, chilling injury is quite a common physiological disorders for the post-harvest handling of fruits and vegetables. Because why? Because for the post-handling of fruits and vegetables, we usually keep, even when we buy our vegetables from the supermarket, we tend to keep in our fridge, right? So this is very common technique to maintain the quality of our fruits and vegetables. Yeah? The most effective method to preserve the quality of the crops. Okay, why? Because they retard aging, they slow down the respiration rate of the crops, and as a consequences, and delay the decay of the crops. Nonetheless, if we store them at the improper low storage, and then chilling injury could occur. Instead of we want to maintain the crop quality, we actually destroy the quality of the crops. So this can cause the great economic loss. Okay, susceptibility to the chilling injury and its manifestation is very widely. Different fruits, different variety, different species have different sensitivity to the chilling temperature. So usually, how do we detect the chilling injury? By looking at the symptoms, such as the surface lesions, discoloration, maybe from the green to yellow or brown, water soaking, the rotten, we have like internal breakdown, and finally decay is the most common one. But if you notice this symptom, it might be quite late already to save the crops. It can be badly damaged already. So, and mostly this chilling injury symptom can only be detected if you remove the crops that you store in a low temperature into the warmer temperature. For example, you keep in 4 degrees, maybe it still look good, but let's say you take it out and keep it at a warmer temperature or room temperature for about 1 or 2 hours, and you start to see the damage symptom. So these are the examples of the different communities, and then they have their like appropriate lowest safety temperature that you can keep them. For example, asparagus, minimum you can keep is about 0 to 2 degrees Celsius, and then as you see in the table, pineapple, I think they only can tolerate as low as 7 degrees Celsius. If you keep lower than 7, it will have chilling injury symptoms. Okay, all right. This is what have been studied before. The injury symptom for avocados. If you keep them about 7.5 degrees Celsius, they start to have grayish brown discoloration of the flesh. Because I think for avocado, the lowest temperature you can keep is about, actually 13. If you keep them lower than that, this symptom going to occur. But somehow it still depends on how you handle avocado or where you grow the avocados or what the subspecies is. And then this is this example of the chilling injury symptoms of the cucumber. Pitting is something like a damage. Can't really see here in the slide. Decay, water soak lesion. This is water soak lesion, this one. And then discoloration. Discoloration you see from the green turn into the yellowish. And then eggplant. Most the symptom for eggplant is browning, especially the seed. And then pitting as well. Okay, for papaya, what are the symptoms? You have like skin skull, hard lumps in the pulp around the vascular bundles. This is the hard pulp. It's not going to be nice to eat because you notice you're going to taste something like hard. It's not going to be soft and smooth. And then they have abnormal ripening, meaning it will not ripen at all. Actually, you will see you store in the low temperature, the chilling injury temperature. When you take up from that low temperature, instead of you expect them to turn from green to orange, yellow, orange, it's not going to happen. This is what we call abnormal ripening. And then finally the post-harvest decay with the growth of the secondary infection from the fungus and bacteria. Okay, mechanisms. For the chilling injury, there are two main theories regarding the onset of the chilling injury. Number one was the effect on the lipid compositions of the membrane, meaning the effect the membrane, the hypolipid of the membrane of the fruit and vegetable. Excuse me. And the second theory is the effect on the antioxidant system, meaning they have something to do with the radicals or the oxygen species, reactive oxygen species. So let's see the first theory. Okay, the first theory is talking about the membrane of the cells of the fruits and vegetable. You see the membrane is in the normal state is a liquid crystal. If you store the fruits in the low chilling temperature, the cell membrane start to change. This is what we call the transition phase from the fluids to the solid. It becomes the solid gels. So what happens is going to disturb whatever the enzyme brown membranes that we are used to transport all the solutes and then big molecules. Is going to disturb this one and especially going to disturb the ATP production of the cells. And then if you keep them for the brief exposure to the low temperature, and then you return the fruits and vegetable to the normal warm temperature, it's going to be okay. But let's say you store them very long, and then this cannot be recovered anymore. The solid gels cannot be recovered anymore. And then it's going to lead to the injury and cell death. This is the first theory. Okay, you relate them with the membrane change of the membrane phase. For the second mechanism, with Sharon et al. 1994 noticed that lipid changes is not the cause of the chilling injury. But what the Sharon et al. Noticed is due to the activated phospholipid catabolism, meaning the attack to the phospholipid structure. So Sheffield and Rosario in 2000, they said that the role of antioxidant to protect the membrane lipid from the lipid peroxidation. And through this lipid production is caused by the reactive oxygen species damage, such as hydrogen peroxide, super NO2 anion, what else? Hydroxyl radicals that damage the membrane lipid and other cellular components. Okay, for this mechanism too, what happens is when you expose the crops to the chilling stress or the low chilling temperature, it's gonna produce reactive oxygen species such as the superoxide anion, hydrogen peroxide, and hydroxyl radicals. The accumulation of these reactive ROS, reactive oxygen species, going to cause the oxidative damage to the lipids, protein, and DNA membranes. And finally, it's gonna cause the membrane breakdown and show the visible symptoms. But with the presence of antioxidants defense mechanisms such as the vitamin C, ascorbic acid, or vitamin E, beta carotene, or those antioxidants enzyme activities such as superoxide dismutase, catalase, peroxidase, is going to scavenging these ROS to maintain the low level for them not to cause the damage to the membrane. So in this one, I would like to share with my research, whereby we just want to see the mechanisms of the chilling injury in okra. As we know, okra is a tropical origin and is susceptible to the chilling injury. So the lowest minimum safe temperature to store okra is about 10 to 7 degrees Celsius. But what happened in our fridge? If we go to supermarket, we buy okra, we tend to keep in our fridge, right? And our fridge, the temperature is about 4, like that, 4 to 8. If you keep the temperature vegetable box, it might be about 8 or 10. But usually it's about lower than that, lower than 10. So what we observe, the chilling injury symptom in okra is you see this as a cell death and the pitting in the pots. When we open up we open up the pot, and then we observe that there's a browning or blackening of the okra seed. And then we also observe the browning of the middle part of the okra pot, peduncle. And then we actually scan this okra seed using a scanning electron micrograph. And then we notice the membrane damage of the seed. With the naked eye, we cannot see, maybe it's just the changes of the color, but what we notice is there's a damage on the surface of the seed, and then the changes of the membrane of the seed. And it's very obvious, especially for the pericarp, the skin of the okra. We notice the membrane thickening compared to the one that safe in the 13 degrees Celsius. This one we do 13 degrees Celsius, this one we do 4 degrees Celsius. So then we also check for the malon dialdehyde. This one is the final product of the lipid peroxidation. So this is quite high for those for the okras stored at the 4 degrees Celsius, as well as for the seeds. Okay, and then this high MDA is to indicate the chilling injury. Okay, and then we also check for the hydrogen peroxide, both in pericarp and the seed. Also very high to those that show the symptom of chilling injury that being kept in the 4 degrees Celsius. Okay, high hydrogen peroxide content in the PMDC indicate reactive oxygen species contributed to the oxidative damage of the okra pod. And then we also check for the enzyme, the antioxidant enzymes such as the peroxidase, POD, and then superoxide anion, dismutase, SOD. And you can see here those okra pod that kept at the chilling injury temperature at 4 degrees has a very low these antioxidants enzymes compared to those that do not show the chilling injury symptom that being stored at the 13 degrees Celsius. So this enzyme SOD, what this enzyme do is actually to converted superoxide anion radicals into the hydrogen peroxide. And this hydrogen peroxide supposedly is being further converted into the water by POD. And then catalase also quite low for those. See, I would not say low, it's quite high for the pericarp at initial storage day. And then it start lowering down as the temperature storage prolong. And then for the seed is high. Why is this in the seed is high? Because there's a studies found that the catalase enzyme activity in water stress sunflower seed and chilling sensitive immature cucumber. This is to induced by the water loss or stress. Okay, for this the first part of the study, the mechanism, we found that those okra pod that exhibit the symptom of chilling injury has high MDA, high hydrogen peroxide, and low antioxidant activity. And then we also, I didn't show the in the graph, but it also has high Browning enzyme activity, PPO. So elevations of the chilling injury by post-harvest treatment. There are many treatments have been already reported. They are categorized in two groups. The first group is physical treatment, such as the temperature treatment, control and modify atmosphere or edible coating. Or we can use chemicals such as the very famous one, 1-methylcyclopropene, 1-MCP, abscisic acid, salicylic and jasmonic acid, polyamines, nitric oxide, 6-benzoyl aminochlorine. So in this slide, I'm going to share with you for my study. This is consisted of the temperature treatment and then polyamine. For the low temperature conditioning or shortcut, we call it the LWT, LTC, sorry, excuse me, LTC. So what happened in this treatment, we actually store the okra pod, condition the okra pod first at the 13 degrees Celsius for about 24 to 38 hours. Then only we move the okra pod to the chilling stress temperature at 4 degrees Celsius. So this one, we found that those okra pod that being conditioned first at the 13 degree has the lower or not showing any symptom at all for the first 8 days. It started only to show symptom after the 12 days and 16 days of storage. Same goes to the weight loss. It has a lower weight loss compared to the control. The control is the one that, the pot that we directly store at 40 Celsius. And then we also notice the symptoms of the seeds. Okay, for those that being treated, low temperature conditioning has almost no changes in the seed color compared to the initial day 0. The browning enzyme activity also quite low compared to the control group. And then we check the MDA. The MDA also lower in the both in pericarp and seeds of the conditioned okra pot. And then we also check for the superoxide anion and then hydrogen peroxide, also lower to those that being treated at the low temperature conditioning. And then we also check for the total antioxidant activity of the DPPH scavenging activity and the total flavonoid. The results show that the treated okra pot has higher antioxidants activity and antioxidant compound. And then we also check for the enzymes, antioxidant enzyme, catalase and SOD also has a higher antioxidant enzyme activity compared to the control, both in the pericarp and seeds. Okay, for the polyamine. Polyamine, we actually tested both, not both, for all the polyamine, the three main one, the putrescine, spermidine, and spermine. This one, if you check at the chilling injury CI index graph results, we actually check for 3, and then this preliminary results show that the 2 millimolar putrescine showing the best, giving the best visual of the okra pot, and then it has also the lowest browning index of the seeds, and then the low browning enzyme of PPO activity. Why is it putrescine? Because it's been postulated or been claimed that this putrescine can stabilize the cell membrane. It has the antioxidant properties, and it also can synthesize stress tolerant compounds such as GABA. Okay, as you look at the result here, you look at the photo of the seeds. This is the control. Of course, the control, this is at the day 16, the last day of the storage. Yeah, the control seeds show the browning symptoms, while for the 2 millimolar putrescine, show just a little bit better than the control. But if you remember the slide of the low temperature conditioning, it has a better visuals of the seeds. So actually it's better. Because for the low temperature conditioning, it can prevent the chilling injury up to 8 days. But for the putrescine, it's only up to 6 days. And then we also check for the enzymes, antioxidant enzyme, catalase, POD, and SOD is higher compared to those non-treated with putrescine. Okay, all right. I'm trying to make it short. I think everybody's hungry. It's almost afternoon, 12:30 already. I mean, here in Malaysia. So this is a conclusion slide. Chilling sensitive horticultural crop. When you're during cold storage, you're gonna produce higher reactive oxidant species, and then has a lower antioxidants compound and or enzyme activity. And then some even say there's a transition phase from the fluid to the solid phase. Okay. And then it causes the chilling injury symptoms. However, with the post-harvest treatment such as now that I show you, the heat treatment or the low temperature treatment or polyamines, it can actually induce the antioxidants, and then this antioxidant will scavenging the ROS to maintain the lower levels, and then it will cause the chilling tolerance. All right, thank you for your attention. Terima kasih banyak-banyak. Please feel free to ask me and try my best to answer.
Okay, terima kasih Dr. Sulika. Please, your home, Dr. Sulika. Well, okay,
sorry, I can hear you.
The presentation for all your speakers. Now we will open discussion session with questions and answers. Yeah, from Ibu J.
Satu lagi siapa ya?
Yeah. So we have a question from Ibu J. Okay, thank you. I think I also also share new insights for me as I'm a traveler. Here I just want to discuss it to Dr. Suisa because I think the chilling injury is so interesting for me. And I only know that something will become that in top leaders, stuff like my favorite or drug like that. It will be the image of the exposure to the health. Yeah, I mean, it is interesting that it can happen for some vegetable and fruits also that exposure to cold leader. I just know that too. But I think all plants can adapt to the environment. Gen will be mutated so phenotype will be shown as the adaptation mechanism. So it may be a choice, the choice for the industry to not only export or import the fruits or vegetables, but also do genetic engineering begin from the time of cultivation. Okay, instead of that, because like what I said the first, then I'm a traveler. I think I want to bring some points and my supply to bring to other countries to save my money when I'm traveling. So, what is the best and cheapest way you think to understand the chilling injury so it can be applicable for all? Such for me that really like to travel and bring some fruits and vegetables to cold weather countries. Thank you. Okay. Thank you, DJ.
Is that a question directed to me? Sorry.
Yeah, maybe Dr. Suisa will answer the question. What is the best solutions that
Yeah, but
chilling injury for traveler?
What's the best? Okay, sorry, I need to repeat the question. What is the best solution to prevent chilling injury? Is that the questions?
Yes, to anticipate the chilling injury for traveler especially.
Oh, for first harvest produce, is there any specific
Yes.
produce that you would like to know, or just any produce in general?
No, actually, I will like.
If for the post harvest, sorry, because it's not that it's not that clear. Yeah.
Coba ditanyakan suaranya jelas. Kita jelas.
Okay, for the post harvest part, to prevent chilling injury is yeah, because the question is not clear. Can you help me if? Okay, just trying to answer just for the sake of answering. For the post harvest handling of the hatchery.
Oh, ada nge-lag gitu kayak kita ngomong dengarnya dulu. Can you hear me? Coba tanya dulu.
Dr. Suisa, can you hear our voice clearly?
Yeah, but it's not that clear. It's not that clear. But what I got the question. Yeah, if possible to type as well. But for the moment, I got the question is how to prevent chilling injury for the horticultural crops. I think the important thing is to know the which is the safest lowest temperature that you can store the crops. For example, for the okra pots, the lowest safest temperature is 10 degrees Celsius. So try not to store them lower than that. But if you happen to store them together with other produce, the best treatment for the moment commercially is 1 MCP, 1 methyl cyclopropene, because it's also can prevent the ethylene. Because ethylene can enhance the chilling sensitivity to the crops. Am I answering? I hope you can hear me.
Yes, yes, maybe it's only short for. Yeah, I think that it will be effective for industrial treatment, like using properly like excellent like that. For supply only
Storage package.
some citizen I mean, like they want to bring it to other countries as for travel or maybe some occasion, and then to prevent that will be biggest. What the best anticipate and the best or cheapest way to do that.
This is challenging. If you happen to travel and then you have to keep everything at one same temperature, and I think 1 MCP is not like everybody can just use it because it's the treatment itself is quite taking some time. I think the best is a modified atmosphere treatment, whereby you try to wrap them. You modify the concentration of the packaging of the oxygen content and the carbon dioxide content to somehow inhibit ethylene as well, that produce ethylene. Because ethylene can enhance, as I mentioned again, can enhance the chilling sensitivity of the produce. So I think for the practical, normal traveling part, packing, you wrap them in the plastic. But you might have certain type, maybe some scrap have a preferable certain amounts of the oxygen and carbon dioxide. But I think the best is to just pack them in the plastic bag and have some holes so that it won't go under anaerobic respiration and then it will kind of ferment and produce alcohol. Yeah, packaging.
Okay, this is not very good. Okay, thank you, Dr. Teresa. Very nice questions for another keynote speaker. Yes. We can come at one here. Come on. I'm so sorry to call me. Okay. I would like to give an ovation to Professor Sensei Takuyama Subramanian. Sensei, watashi wa Ika desu. I would like to ask a question because your presentation is very interesting to me. And maybe I have first questions about the chemical compound of nobylate. Is the chemical compound of nobylate is especially found in the field of the citrus, or we can find it in the fresh fruit? And secondly, are there any products that has been which has been commercially in market, especially in Japan market? Thank you. I hope you understand what about my questions. Okay. Okay, Professor Akulia, do you hear our voice?
So sorry, I can't catch you. The sound is not,
Okay.
quality is not so good. I
Yeah,
cannot
you
find.
will repeat again the question because. Okay. Sorry, Mas Say. My question is about mobility compound. Is the mobility compound is special that you have mentioned is that we can found only in the pure citrus like your research, or we can find in the flesh citrus. Things like that. Do you have any information about?
Could you mention in the chat? I can't hear you. I can't hear clearly. Sorry.
Oh, gitu.
Sorry. Could you mention in the chat?
Saya bantu. Tapi ke sini. Lebih jelas. Mungkin lebih jelas kalau di sini. Kalau agak jauh kayaknya kurang jelas.
Your term or your time about your subjects, your research is about mobility compound, right? So, Sensei.
Yeah. No question. Yeah.
Yes.
Compound from the citrus.
It is found. It is only found. We can find that mobility compound is only in the peel of citrus, or we can find mobility.
Yeah, mainly in citrus. Mainly in citrus. Yeah. However, I don't know what kind of products. I don't know, but mainly containing in citrus. Yeah.
And it is only in the peel of the citrus, right?
Only pear, only
Yeah.
mainly pear. Citrus pear, yeah.
Yeah. Only in the peel of the citrus, not in the flesh.
Yeah.
Yeah.
Little amount in fruits and in the fresh, yeah.
Yeah.
Mainly in pear,
Yeah.
outer pear, yeah.
It means that in the peel we can found the chemical compound of mobility is most good abundant from others chemical compound, right?
Sorry, I can't catch you.
Yeah. And about the horseshoe, is there any other trend about the horseshoe for food functional products in Japan? We are so interested. Yeah, about the trend of the food regulation about the food functional in Japan.
Yes. Also citrus and some other agricultural crops and some fermented food also have some function, immunostimulation, anti-allergy, and so many substances in some agricultural crops and many substances. We are studying about the many substances and some medicinal plants also have some substances, flavonoids and carotenoids. And then because we are focusing on the citrus, because the citrus is very, very major products in Ehime Prefecture in Japan. So other products like fish also have the many functions, and DHA and EPA, the very important fish oil, also the anti-obesity activity, something like that. So this is the reason why we are evaluating so many kinds of products. Yeah. Okay. Is okay? Sorry.
Okay.
Okay, it's okay, Professor.
Thank you, Professor.
Thank you very much, Professor, for the answer. Thank you, Dika.
Thank you. Thank you.
Okay. Do we have another question for the female speakers? Yes.
Yang angkat tangan.
I will ask to Prof. Sugara, is there any different effect from orange skin extract from different variety?
You mean there are so many kinds of stresses in Japan and maybe in Indonesia also. So each stress species containing the contents in stress is quite different from each species. So the one species mainly containing beta cryptoxanthin, the carotenoids. And then some other species containing some coumarins or leptin in some specific species. So yes, each component quite different between the stress species. So we are evaluating each evaluating function and evaluating the components in each stress. And then we are focused on the major component in stress. Yeah, quite different. My answer is quite different between the stress species.
All right, thank you, Professor.
Okay, thank you very much, Professor, for the answers. Thank you all. Yeah. Okay. Yeah. We will have questions from Zoom. Bapak Gufron Action.
Ya, halo. Halo, apakah sudah bisa kedengaran suaranya?
Ya, sudah bisa, Pak?
Oh ya, langsung aja, Mbak ya. I have question for Dr. Samsul Haq. It's about what kind of mycorrhizal blend did you use, or probably commercial or laboratory product? And the second, hello, sorry.
Yes, Pak.
Oh yeah. What is your hypothesis about probably you can add full combination with biochar, mycorrhiza, and fertilizer, and what is your hypothesis about that? And for the total dry matter, it is include harvest product or only plant part of dry matter? And the fourth question is about cellulotic bacteria. It is from the biochar, I'm sorry, bacterial consortium or from the mycorrhizal fungi activities. And for Dr. Sohaib Mazhar, I have question. What is your recommendation of antibiotic use in livestock practices? And what is your suggestion of the best practices to replace antibiotic? That's my question. Thank you.
Okay, thank you, Pak Gufron. Okay, please, Dr. Shamsul.
Yeah, no, thank you. Yeah, so far I noted four points. See if I can miss anything. Mycorrhizal blend. In Australia, we only got one commercial company who actually selling commercially mycorrhizal fungi, but not the bacteria. So the bacterial one, we can say, like how I get it, I actually work in a industry who actually working on that one, and they are believing in next 3 to 4 years they will commercialize that one. So I actually get that bacillus one from them, but the mycorrhizal one you can get it commercially. That's one thing. The second question was my hypothesis that when you are combined all of the factors, then what will happen? So my it's difficult for me to make a concluding remark, because one of the limitation, because this trial we just did our lysimeter, which is not 100% to our field condition. So the main problem is when you put this treatment in the field condition, so the soil water actually help leach down many of the materials. So it's quite different than what we are actually doing in a confined environment. The limitation is in the field condition is difficult to get the soil sample, for example, 50 centimeter below. So we are actually working on that one, and we try to almost develop a method where we actually slice 1 meter of profile, and then we put a tube underneath the column so that the flushing of water actually coming through and we can pick it from there. So we just developed that one. I'm planning to set up a trial next year, and we try to collect the soil pore water, and we then analyze, and then maybe we can answer that one, that whether it is effective or not. Your other question is the total dry matter. Yes, so total dry matter, that above ground stem, including leaf and panicle, like the head. This is, I say, total. And when I ratio that one to the just grain, then we calculated harvest index. So that is your point. The other thing is that this cellulotic bacteria, is it microbial like the fungi or not? Yeah, this is from fungi. So we got that one only for fungal treatment. It will be really interesting if we can isolate those microbes, put it in a treatment, and then if we add bacillus, and we can see that whether it is antagonistic each other or friend to each other, and whether it is beneficial or not. The problem is, you know, that in a soil we got thousands of species of microbes. Now, the limitation is when we are culturing a particular microbes, we don't have others actually sitting. So we cannot actually differentiate that whether they will do in 3 or 4 different microbes together, are they behaving the same way or are they behaving a different way? That's one aspect. The second one is when you get this type of microbes, how plant root actually reacts of that one. When you are talking about root exudates, what actually they are signaling, that they signal the microbes to grow more or less. So I think this is something, the research topic for next 5-10 years, that how we can actually sort it out this phase. I'm not sure, did I answer your question?
I think it's okay.
Okay.
Thank you.
Thank you. Yeah. Yeah. Anytime, you got my email. So if you if you are not clarified, you can just send me an email. We can discuss further. Okay. Cool.
Thank you.
Thank you.
You, brother.
We cannot hear you.
I think I get the point from your answer.
Okay. Okay. No, that's fine. No,
I'm happy.
You're fine. But I think the in the stage, I think I cannot hear.
Oh yeah. There is no response.
No, I think she's talking, but I cannot hear
Oh,
the lovely voice. No.
yeah, I have question for Dr. Sohaib Mazhar, I think.
Yeah, okay. Dr. Shoaib, he is here, I think. No, I cannot. I'm not sure it's in another page.
Yeah.
Sorry, the icon first to screen. We cannot hear you at all. No, unfortunately not. I can see that you are talking something, but I cannot hear your voice.
Yes.
Dijawabkan
Coba di ini lagi di
Ya.
sini ucapan terima kasih.
pertanyaan tadi.
Pertanyaan sebentar. Jadi udah dijawab ya.
Any question from the participant on the Zoom, maybe? Okay, thank you, everybody, for the questions. Yeah. So if we don't have any questions, maybe it's over the session for the keynote speakers. We do hope that all the keynote speakers will share the slides to the committee, right? So maybe we can learn in detail about the slides from the keynote speakers. Except for Professor Takuya is already sent the slides to the committee. Maybe we will wait for another 3 speakers to share the slides of the presentation. Okay, if we don't have any other questions or comments, maybe we can end this session with take the picture together. Okay, maybe for the participants from the Zoom, maybe you can turn on your videos on your camera. We will take the picture with the keynote speakers. Maybe from the first slides first. Okay.
First slide gimana?
First slide.
Siapa?
Yang slide siapa?
Dari slide yang basket.
Yang tadi.
Yang pertama.
Yang pertama.
Enggak, betul itu yang dari slide yang pertama. Bapak screenshot yang pertama ini. Tanya pertama.
Okay, 1, 2, keys.
Sekali ulang.
Okay.
Ini lagi.
One more.
Ini.
1, 2, keys. Okay.
Second another slides.
Ulang lagi. Bisa diulang lagi enggak?
Okay. One more. 1, 2, keys.
Oke, udah ada di tersequence.
Okay. I think this is all for our presentation today. Thank you so much for all the keynote speakers for the wonderful presentation, wonderful experience. We hope that we will have the next agenda or the next conference. All be the second international conference, and I can pass in the next year. Ibu Jaya, you do so. Thank you so much for all the participants from the Zoom and from the accounting hall and stay connected in our channel. Give applause for all the keynote speakers today. Also, thank you so much for all the Everyone who gave the questions to the keynote speakers. The last, I am Lulu Fadila. Thank you so much for the opportunity it has given to me. That after this, the event will be held by the master of ceremony. Thank you so much. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Well, thank you so much. Can you give applause for our keynote speakers and please, Lulu Fadila. SVG, LEG, please forward.
Thank you.
Well, dear ladies and gentlemen, thank you for your interest here and in the Zoom. It was really interesting session, right? I like the material, I like the presentation, and I want to say thank you for our keynotes, our Excellency Professor Taufik Subahana, Dr. Samsul Haq, Hafiz Sohaib Mazhar PhD, and Dr. Teresa Pile. Give applause for our keynotes. Well, a very big thank you to all the guests who have spoken with us today. Finally, I would like to wrap up with some of the committee notes for your attention. Firstly, there will be an hour break for lunch, prayer and preparation. So we will begin our next agenda at 1 p.m. Indonesian time. And I want to say thank you. Secondly, the parallel session will take place hybrid at here and Zoom. The name of speaker and each classes has been taken at the ground of this room and share on email as well. So please arrive punctually. Since the next session will be led by each moderators of parallel session, I would like to say one. I apologize if there are any mistakes in leading this agenda. I'm so sorry if I'm not here today. I'm Adele, Master of Ceremony on this event, on this conference. I want to say thank you for our guests in here. Come to the council hall, and I want to say big thanks for the wonderful, the great, our lecturer, Ms. Nurul and Ms. Jenny. They are so great, and my beautiful lecturer, of course. So, and I wish we can still meet again in the next conference. Wassalamualaikum warahmatullahi wabarakatuh. Shalom and see you later. Goodbye.
Thank you.
Apa yang untuk sesi berikutnya?
Oke, baiklah mungkin sekarang juga kita break, kita akan istirahat dulu. Lunch break. Ya boleh, mungkin nanti juga ada yang mau sholat. Langsung kita silakan. Yang panitia juga semuanya. Kita ada di belakang Pak, langsung jadi silakan diambil.
Langsung saya ini ini. Siapa yang harus kunci itulah kurang sebenarnya. Pak saya ya Pak saya siapa ya share screen itu